All right, let's dive back into electrical properties of neurons or the action potential and finish up a little bit of what we didn't get to cover last class. All right, so I thought it would maybe be a good idea to take just a few seconds here, take a step back and think about what should you actually know. Now, there's a lot of events that occur in the nerve or in the action potential, right? Ions moving, channels opening. We talk through all of that, and you should understand that, right? You don't necessarily have to memorize it. I think it would be a little useful to at least have memorized the basics, but you should understand them. And then more to that is think about what would happen if you could alter these or change these, right? This is that sort of higher level of knowledge. What happens if you sort of perturb the action potential? What's going to happen? And this will become more interesting, hopefully in a little bit here when we get to the idea of drugs, right? Because in my mind, what would be the most useful for a lot of people is to understand what happens when we alter this, right? And most drugs that affect the nervous system are going to affect either the action potential that we're talking about now or the synapse. And that's why understanding the action potential and the targets of those drugs can be useful. All right, so you hopefully have seen this video too many times now. But to get this idea, right, the action potential is this emanating blast that's going down the dendrites, going to go out the axon, and we can see this rapid depolarization or increase in voltage that goes right back, and then we can fire it on the potential, right? So that's the gist of it. Now let's talk about some things we haven't gotten to yet. When I asked you for the worksheet to draw out an axon, we talked about myelin, right? And we've talked about Schwann cells and oligodendrocytes and how they can wrap the axon and myelinate it. And we said this is good for the action potential. It makes it more reliable and it makes it faster. But how is that so? And the sort of base level explanation is the idea that the action potential can sort of skip over these areas from node to node, as these are going to be called nodes of Ranvier, in between the myelin, and that allows us to go faster. It also means that it requires less energy to do so, and overall this should be good. Now, we will get to a slightly more truthful and realistic, but also complicated definition in a bit here. To show you a picture, you can actually see these, right? So this is an example of a neuron, and remember we talked about immunofluorescence when we went over the paper? But what you can see here is if you stain for the sodium channels and for the potassium channels, the sodium channels are red and the potassium channels are green, we can actually label right where they are. So this whole stretch has been myelin, and then right here they put a whole bunch of sodium and potassium channels right at these nodes. And this is a great sort of energy saving to the cell because we don't have to put them all along here. Now I think this sort of shows a little bit better, and I will go over this in class a little again because I think it is complicated. Why does this actually make a difference, right? And so in here it's trying to show these sort of yellow areas here are the myelin sheaths. And the main idea is you just put all your channels right at these nodes in between, and then the ions can diffuse much more quickly between them. Now, why can they diffuse more quickly? Again, this is a little bit hard to show. Let me see if I can get a tool up here to... And, right? So we think about it. We have all of these right at rest. We have a whole bunch of the cell is more positive outside, more negative inside, and that's what causes sodium to rush in. However, if we have these two different charges, imagine this, right? If you have two different magnets, right, and they have a different charge to them, you know that we got like the two different poles in there, they stick to each other, right? And it sort of works like this. If you had a positive out here and a negative out here, they're going to act like those magnets, and they're going to clamp right on to this part of membrane here, and they're not going to want to move. It's like those magnets stuck to each other. However, like we know this too, if we ever play with magnets, if you hold them right next to each other, they're hard to pull apart, but you hold them, a little bit further apart, and you don't feel any force at all. So if you put a magnet right here, a positive, and a negative here, they don't affect each other very much. So now these are much more able to flow because they aren't under the influence of this sort of external charge. And that's the real key to how myelination makes things go faster. This makes these ions flow very quickly from here to here and allows that action potential to sort of jump from spot to spot, or seemingly so, right? It's not actually jumping. The ions are going to, right, remember what has to happen here is that for these sodium channels at the next node to go off, we need to get a little bit elevation in that voltage. So as the sodium flow in here, then they're able to sort of quickly flow down to here, and then that opens these next channels. All right. I'm going to re-bring this up here, but this is again showing the sodium-potassium pump to show after the action potential. You get this happening here where this pump is able to put things sort of against their gradient, right? And it takes energy to do this. That's the real key of this. Whereas opening a channel, you just open, things freely go through. This is much more like, let's use an analogy here, right? The channel is like you have a dam and you open a hole in the dam, right? The water is going to flow down into the part with less water, right? But a pump is like if you have a well, right? The well doesn't want to come out of the ground. You need to, either pump the handle or add in some electrical pump that's going to pump it out. And that's the idea here. We use ATP in biology. That causes this thing to really act like a pump where it starts putting 3 sodium out, 2 potassium in, 3 sodium out, 2 potassium in this pump-like fashion. All right, we aren't going to spend too much time talking about this now, but I do want to bring this up, right? So this is a graph of voltage over time again, but it looks a little different than we're used to, right? This, if we remember right, we said there's lots of different sodium and potassium channels, and this is an example of a neuron that once you start it going, it becomes bursting, as we would call it, right? It just keeps firing action potentials constantly until you actually turn it off, right? You bring it back down. So this is one of the things that can cause a neuron to behave this way. And this is a normal thing. This isn't necessarily a bad thing. We have a lot of bursting neurons, and they're very important for keeping things regulated in our brain. What do you think you could change about the properties of the sodium channel that would cause it to do this? Right? I'm going to encourage you to pause the video and take a couple minutes and write down an idea, because we'll come back to this in class next time. All right. So we mentioned that neurons are all or nothing in regards to the action potential. Either you fire a full action potential with that full spiking, or you see nothing. I do want to mention this again in sort of all honesty. There are a few exceptions, like always, of neurons that are graded potentials that can have, right? If we say this is the normal action potential, you can get neurons that do have these sort of, right? It can do that, or it can do this, or it could do this, right? Tend to be a little bit slower and have these graded potentials. And they also, because of this, they decay as they go along, right? They don't maintain that all or nothing thing. So if you're very close to the start of the graded potential, you'll see its full effect. But if you are, hundreds of microns away, it might have a very minimal effect. We aren't really going to talk about these after this fact, but it's just one of those things that I think is worth knowing that an exception exists. They're often most important in sort of your retina as well, is where I usually put them. All right. So we talked about sodium, or about voltage-gated channels. Let's talk a little bit about other ways channels can be gated that will actually start how this works, right? So this is an example of what happens in your inner ear, right, where you're actually sensing sound waves. And there's a whole bunch of things which I think might come out later, but essentially sound is going to come in through your ear, go through your eardrum, it'll vibrate some fluid, and then it's going to vibrate, right, because sound is really just vibration, this tectoral membrane, right? And this tectoral membrane is then attached to what are called hair cells, inner ear hair cells in particular. There are a few different sets of these. This one right here are actually the important ones for sound. What they do is they're these cells, they're actually a type of neuron, and at the end they have these tiny, tiny little hairs. And these are connected here, and so if these vibrations move, these little hairs move, right? And these hairs are actually, the movement of these hairs is what actually causes channels to open that cause action potentials that give us the sense of hearing. So here's a little video to show that example. So here's these little hairs, and they're just showing that they can literally deflect them sort of back and forth. These hairs are called stereocilia. It'll probably look better if you look at these after the fact. All right, so here's the kind of neat thing about how these work. At the ends, at the very tips of these, right, there are these tip link proteins and channels. So there's a channel here that's shown in red and a little molecular fiber made of protein that connects it here. So basically, if you move this now this way, right, you're going to sort of stretch or pull at this, and that literally opens up this channel and causes ions to rush it, right? So it's positive charge, just like sodium, and that can in turn lead to an action potential, right? Another example, so in neuroscience, there's a lot of times we want to be able to decide when a neuron's on or off, right? So there's been a lot of ways that we figured out how to do this, right? The sort of oldest way I can think of are these shaker voltage channels that were found in flies, I think in the 70s, right? And what they found here is that this is a calcium channel, right? It's a big positive ion. And if they put the flies at one temperature versus other, they realized that they could open and close these channels. So then if you put these channels, you decide, I want this to be in this specific neuron, right? You can see what happens when that neuron's inactive, or you put it at a different temperature, and you can realize, oh, that's what it looks like when they're active. So you can, in a living animal, you can suddenly turn on and off specific sets of neurons. We talked about this earlier on, and we'll talk about this later. This is sort of the more cutting-edge way to do that, and that's using optogenetics. Where again, now you can put into a specific type of neuron a sodium channel that is gated by light. You can, if you hit it with this specific blue light, you open it up, right? So now we get even more, right? We can decide which neurons, and we don't have to do this temperature trick anymore. We just have to run a fiber optic cable into a brain and decide which they turn it on, right? So that's the idea here. Now we can hit them with light and open up this channel. Also, a more recent technique that is becoming increasingly used is that they make a whole range of designer channels. This can be all sorts of different types, right? They can turn neurons on, off. They can sort of modulate them to sort of slightly turn them up, make it more likely without actually turning them on, or slightly dial them down. But anyways, the thing about these is that these are work by use of a very specific drug that you can give. This drug doesn't do anything else unless this channel exists. So now you can decide, I want to put this channel in this specific neuron of this mouse, right? That doesn't do anything. But if I give them this particular drug in their food, now I've altered that channel. You can see, does that cause changes to the behavior, changes to other neurons, and so forth. Okay? All right. So that's, I think they're basically wrapping up the action potential for now. We'll come back more and work through some problems with to help you sort of try and better understand how the action potential works and how we can perturb it next class. But I think we've covered the basics well enough.